some people. Yeah. Hello, everybody. Thank you so much to join this webinar. So this is the first one we are thinking to do in a series of webinar related to the several aspects in the ship industry. Today we are going to talk about how genomics can help you to increase the profitability of your flock. For those that you don't know me, I'm Angela Canovas, the still new professor in University of Guelph for beef genomics and small ruminants. And today I have a pleasure to introduce our speakers for today. The first one is Dr. Luis Brito. He has an honors bachelor degree in animal science in 2010 and in Master of Science in 2012 in genetics and animal breeding from the Federal University of Viçosa in Brazil. Luis Brito completed his PhD recently in 2016 at the University of Guelph, here in our group under the supervision of Professor Flavio Schenkel. His thesis title was Genetics and Genomics in Small Ruminants, and he conducted part of his PhD research in the Invermay Agriculture Centre in Agri Research Institute in New Zealand and at the Cesairo Research Institute in Australia. In September 2016, he started working as a postdoctoral researcher at the Center of Genetic Improvement in Livestock here in our group in University of Guelph. Currently, Luis is connected from France. He is doing an internship in INRA, is a research institute in France, working, well, in several research projects, but more focused on establishing an international collaboration to do a genetic evaluations for goats, and also what is there working on sheep. So thank you so much, Luis. We appreciate a lot that you are connected from France in this time that is later in Europe. So thank you so much, and yeah, I hope you enjoy his presentation. When Luis is going to finish, Delma Kennedy, the sheep specialist in OMAFRA, is going to talk also about genomics in the sheep industry. So thank you so much.
Okay, thank you very much for the introduction, Angela. And hello, everyone. And it's a pleasure to talk to you here today about sheep genomics. So as Professor Angela mentioned, so I have done quite some work in the area of sheep and goats genomics. And so today I want to share with you some of my passion and also what I have learned about genomics. And I have seen that investing in genetics can be very advantageous for several livestock industries. And there are numerous publications and reports of great genetic progress done for those industries. And in Canada, for the sheep industry, we are a little bit far behind compared to other countries in the area of genetics, of sheep genetics. And so today, during this presentation, I want to show you and discuss with you how genomics can help you to increase the profitability of your flock. So we are going to discuss some points, what you need to do, how genomics works, and the data that we need to generate to do the genomic evaluations, and what is it, what are the benefits, and how you can implement it in your flock. And at the end of our presentations, we are also going to take some questions, so feel free to ask any question. If you don't understand any concept, please just let us know, okay? And so I want to start with this picture. So when you have a crop of lambs, if you just look at them, are you able to tell which one is going to give you more profit? So which one, maybe you have one that grow faster and grows more, but at the same time could be an animal that eat much more compared to the other ones. Or you could have an animal that get much more disease, that is less resistance to parasites, or that may not produce a very good carcass. So just looking at the animals is not easy to tell which one is going to be the best one. And the same applies when you are choosing which animals are going to be used for breeding. So let's say if you have two males and you need to pick one of them to use as a ram in your flock. If you don't have any data, it's hard for you to decide which one is the best one. Because if you have one that was born, for example, as a single, and the other one was born as a triplet, the environment, so it means that the performance of this animal was affected because it was also sharing, let's say, the milk from its mother, it was sharing with its siblings. So there are several environmental effects that are going to influence the performance of an animal. And however, it's important to know the genetic component of that animal, which is what's going to be passed to its progeny. So the profitability of your flock depends on several factors, such as the facilities and the pastures that you have, depends on the quality of the nutrition, the health condition of the animals, the management practices, reproduction, and there is a big component which is genetics. So you may give the best environment to an animal, but if this animal does not have a good genetics to express um, its potential, this animal is not going to perform well. At the same time, if you have an animal with a very good genetics, but if you don't give good nutrition and if the animal is not housed in very good facilities. So we need to combine all those factors in order to have a very profitable flock. And today, as I mentioned, I'm going to focus on the component genetics. So I want to discuss with you the importance of improving the genetics of your flock. But why genetics is important? So here I just presented, this is one example from chicken, where they had two lines of chicken. On the top, those are the genetics from 1957, and on the bottom, that's the genetics of today. And all those animals, they were fed the same diet, exactly the same diet. And as we can see, at the same age, for example, at day 43, day 57, we can see the difference between the 1957 and the genetics of today. So this shows us that genetics works because the animals were having the same environment, the same nutrition, and the genetics of the bottom, which is the animals that were genetically improved, they perform much better, much bigger carcass, better efficient, feed efficient. So that's why the same can also happen in the sheep. And I'm going to show it that there is already an example of this genetic progress in New Zealand sheep. So this is one report from the group that I spent some time in working with in New Zealand. And I just present some traits. And you can see here that, for example, for lambing percentage, from 1999 to 2006, there was an increase of over 100% for this trait. The same for average lamb weight. There was also almost 20% of increase. Lamb sold kilograms per ewe, also over 70% of increase, and the majority of these gains are due to genetic selection. They have a very structured breeding program, and they have worked very hard to select for the majority of the traits that are going to influence the profitability of the New Zealand sheep producers. And I think we can do the same in Canada. So how genetic selection has been done so far? First, there is the first option is phenotypic selection. Selection is selection that's done basically visually. So you just look at the animals and you decide which ones you are going to choose to be the parents of the next generation. And phenotypic selection is not very efficient because you are not able to distinguish all the environmental effects that are influence the performance of an animal. So you may select an animal for to be ran in your flock, which may be an animal that does not have very good genetics. That animal may just have had very good environmental conditions, while the other one that you could choose did not have the same conditions. And the second approach for genetic evaluation is what we call traditional genetic evaluations, in which we use phenotypic information and also pedigree. And we already have a breeding program in place in Canada, and Delma Kennedy is going to discuss more about this program, how it works, and also how you can join this program and be part of this genetic evaluation. And the other approach to do a selection is called genomic selection. But what is genomic selection? In order to understand genomic selection, we need to understand what are genome markers. Why? Because in order to perform genomic selection, we need to genotype our animals and we need to know their genomic information. So you see here in the first image here, that's the DNA of a sheep. Most of so we can see here that there are different letters, what we call nitrogen bases, and most of them they are similar for all the animals. We have here, for example, on top animal 1 and on bottom animal 2. However, there are some of those markers which are different between animals, and those differences they are going to make make up the genetic difference, the difference that we observe in the phenotypes of an animal. For example, we have a trait such as carcass weight. There are several markers in the genome that are going to influence the expression of this trait. So it's very important that we know that we genotype the animals in order to identify those markers. And as you may hear, those markers, they are called SNP, or also single nucleotide polymorphisms. Why? Because they are some polymorphism in specific regions of the genome, and those variations in the genome are going to cause variation in the phenotypes, which could be heavier carcass weight, which could be animals that are more or less feed efficient, more resilient or resistant to parasites. And how do we identify those markers? So since the sequence of the sheep genome, there has been the development of a technology which is called SNP sheep. That's the second image, where we collect the DNA of the animal, we place in this chip, and then it goes in a genotyping machine, and this is going to generate all the information that are going to be used for genetic evaluation. But how the full process works? So that's what we have here. Let's start from the top here. So the first step, when the animals are born and you are going to tag them to include their electronic ID, so this is one equipment that can be used, the applicator, and we also have the tissue sampler. So when we are going to, or at the same time that we are going to tag the animals, we accopplate this tissue sampler to the applicator, and at the same time we are going to collect a biological sample from the sheep ear. This what we call ear punch. So that's what I show in the second column. This ear punch, the DNA is then taken to a laboratory where the DNA is extracted, and after the DNA is extracted, it is placed the technology in the SNP chip, as I mentioned previously, and it goes to the genotyping machine, and it's going to generate a lot of data. Each point, as you can see here, each point is one marker, or we can also call SNP, and each color here is one chromosome. So we have, as we can see, we have a lot of information from all the chromosomes, which are then going to be responsible for all the genetic variation that we can observe for all the traits that we are interested in. Based on this information generated from the animal DNA, we can then estimate what we call trait prediction equations, which are going to be used to generate the genomic breeding values. And the genomic breeding values is an estimate that you are going to have for each trait and each animal. And based on those values, you are going to choose which animals you are going to use for reproduction and which animals you are going to send for slaughter. And based on these results, you can take more accurate decisions. So you can, instead of just looking at an animal and telling this animal is better than this one, by just looking at the animal, just made visually, so you can have actually a calculation that shows, based on DNA information, the genetic potential of an animal. And what are the opportunities of genomic selection? So I want to discuss which traits are going to be more advantageous when using genomic selection. And those traits are, for instance, sex-limited traits. For example, how do you select a male for milk production? One option is to do progeny testing. So for each, you breed each ram with a variety of ewes, and then you do the measurement in all the progeny, and based on the daughter's production, you are going to select the males. However, this is very it's very expensive to do it, and also take a long time because you need to wait the animal arrive in the reproductive state, and then the animal is going to reproduce, and you have to wait for 5 months, and then you have to wait until the lamb grows and get pregnant and produce milk and finish the lactation, or if you were working with meat sheep until the animal uh, is sent to slaughter. So traits that are sex limited, they can be very advantageous using genomic selection because you can estimate the genetic potential of an animal at birth with a good accuracy. Also, for low heritability traits, such as fertility, and there is a variety of important fertility traits, traits that are also difficult or expensive to measure, such as feed efficiency, fecal egg count as an indicator of um, resistance to parasites, and carcass traits. So all those traits, they are measured at a later age, or they can be expensive to measure. For example, to record feed intake, it needs some equipment. You need to have a control environment where you have control of everything that the animal is eating. And with genomics, if you have a group of animals that are genotype and phenotype, you can then estimate the potential of the young animals, and you can select those animals and make genetic progress. Also, traits that are expressed late in life, such as lifetime productivity of use, or also disease susceptibility, disease resistance. For instance, in New Zealand, they have been using genomic selection to select for resistance to parasites, to pneumonia, to facial eczema, which is a disease very important in the New Zealand dairy shipping industry. So there is a lot of opportunities for genomic selection to help in the breeding program for a variety of traits. And there are also other applications of genomics, such as parentage verification. So let's say if you did not record, or if you, for some reason, a ram jumped the fence, or if you had multiple rams in the same field, and if you don't know which one is the sire, based on the DNA information, you can then detect which one is the sire of the progeny. And this information is going to help you to have a more accurate pedigree, which is going to benefit the genetic evaluation. There are also some genetic abnormalities that can be identified by DNA test. It's also used to identify the genetic diversity of your flock. So it's important, we know that it's important to control inbreeding in our population. So that's why with genomic information, we can more accurately estimate the rate of inbreeding per each animal, and then we can decide which animals we are going to mate in order to generate a progeny that has a very low inbreeding level. And the reason for that is because inbreeding can affect high levels of inbreeding can affect performance traits, survival, fertility, and several other traits. DNA information can also be used for traceability, so to identify the breed, for example, in a slaughterhouse, based on the DNA information, you can identify which breed is that carcass from. So, and you can trace back things from the farm. So genomics is also used for that. And what I have already mentioned, genomic selection. But how genomics increase the genetic gain in sheep? There is one simple and very important formula which says the genetic progress that you make in your population depends on three main factors, which are the selection intensity, which is an indicator of the proportion of animals that you are going to keep to be parents of the next generation. For example, let's say if you have 1,000 lambs. If you choose the top 10 animals, you are going you are putting a much higher selection intensity than if you were choosing 50 animals. Why? Because when you choose 10 from 1000, you are choosing the best ones. When you choose 50, it means that you are being less, you are choosing a greater number of animals. At the same time, you are not being so strict as you were when applying a higher selection intensity. The accuracy of the phenotypes that you collect as well is going to influence the genetic progress. So as we collect data more accurately as possible, this means that the genetic progress is going to happen faster, and also the generation interval. So those three components are the main ones. So we want to increase the selection intensity, we want to increase the accuracy of the phenotypes that we are measuring, and we want to decrease the generation interval. If we do it, we are going to have a greater genetic progress. But how can you do it? First, you need to collect pedigree information, you need to collect phenotypes, and you need to genotype your animals. And it's very important to collect this data in the best way possible. Why? Because when that's why I put this picture here, which says garbage in, garbage out. If we don't collect very good data, for example, pedigree, or if we don't measure the animals, if we are working with carcass traits, so if we don't weight well the carcass, and or if we don't record this information well, it means that the results are not going to be as good as we want. So it's very important that when recording all the information in your flock, that you do it as accurately as possible. So here there is one question: which of the following statements is false? The first one, phenotyping is hard work. This is not false. Of course, this is true. It is not easy to measure, for example, to weight the animals, or it's not easy to measure feed efficiency, or to record number of lambs born, number of lambs weighted, to record when the animal was bred, when the animals gave birth, who were the parents. So it is hard work, but hard work pays off. So it's important to have all this information. The second one, phenotyping is expensive. That's true as well. Why? Because when you are recording data, it means that you are yourself is putting your time to do it, or you are paying someone. And labor is also expensive. So there is a cost to generate this data. And what we have seen from other industries, such as dairy, beef, even sheep in Australia, New Zealand, in France, we can see that the hard work and the investments that they have made to phenotype and to collect this data, they have received it back. Because if you have animals that produce more, that are more resistant to disease, that are more efficient, it saves you money. And so you put some money to record the data, but then you have more animals that have a better genetic potential, and they are going to perform better in your flock. The third one, phenotyping is time consuming. Yes, because you need to record all this information, you need to keep all this information, you need to send this information, for example, to a program such as Genovas to keep track of all this data and to perform the genetic evaluations. Also, when choosing the animals, you are going to use all this information to select what are the best animals that you have in your flock. And the last one, genomics is going to save us from all this phenotyping. And that's the answer. That's the false. Why? Because even though using genomics, it's still needed to record phenotypes. Genomics is going to help, but it's also important to continue recording phenotype, pedigree, and to genotype the animals. So that's the message that I want to give from this slide. Phenotyping is hard work, is expensive, takes time to do it. However, it's very important to have a successful breeding program in place. And that's what we have already observed in several other industries, that they have made a lot of progress by phenotyping, collecting pedigree information, and genotyping their animals. Just to give you here an example, this was one study that was done in Scotland, and I only presented here two traits, which are fecal egg count at 20 and 24 weeks, and they are indicators of parasite resistance. So we can see in the second column that when using only pedigree information, the accuracy was 0.48 for the first trait and 0.41 for the second trait. When they include genomic information, the accuracy increased from 0.48 to 0.60 and 0.41 to 0.50. So this was a great increase in accuracy. And as I previously mentioned here, when we increase the accuracy of the phenotype that we are recording or the breeding value, we are going to also increase the rate of genetic progress in our flock. So that's why genomics is helping in the breeding programs. So in New Zealand, just to give an example of a successful story of breeding program in sheep, that's New Zealand, it's a very successful one, where they have already incorporated genomic selection in their breeding programs for various traits, including feed efficiency, meat quality, parasite resistance, number of lambs born, meat yield and adult ewe live weight, and several other traits. And they are also collecting other phenotypes in order to select for other traits. And so if you collect data, you are going, and if you have the genotypes, you are going then to be able to select for other traits that you may be interested in, traits that are economically important for your production system. And as it has happened in New Zealand, it can also happen here in Canada. And the gains, they are very significant, from 30 to 70%, depending on the trait. And other than genomic selection, they have also developed some major gene tests for some specific traits, such as the Loin-Max and Myo-Max, which are related to select for eye muscle depth or muscularity traits, and also the Worm Star to select for parasite resistance. And there are also other tests that they have developed. And they are also using, as I mentioned, genomic selection to improve these traits as well. During the period that I was in New Zealand, so I worked with a data set of a multi-breed sheep population, and I was working with various growth, carcass, and meat quality traits, and we showed that the accuracy for those traits were very good and show that great genetic progress can be done for those traits if genomic selection is performed. And in Canada, we have a similar structure where also there is a lot of breeds. So it's important that we develop methods to implement genomic selection in sheep here in Canada as well. And there are technologies we have at the University of Guelph, at Omafra. So we have the tools, we have the experience to do this work. And lastly, I want to bring your attention to another study, which is transcriptomics, which is another type of study where we, let's say, we have two group of animals. One, animals that have a very low fecal egg count, which means they are more resistant to parasites, and we have another group which have a very high fecal egg count, which means that they are very susceptible to parasite infection. And we can have a better biological understanding of what are the genes involved in the trait expression. So what we can do is we collect biological material from both groups, and based on those biological materials, we are going to compare what are the genes that are expressed in the low and in the high fecal egg count animals to then identify genes that are associated with parasite resistance. And the same study can be applied to any other trait. Okay, so this is the first message that I want to give you, that gains have been made in Canada, and Elm is going to talk more to you about Genovis. Genomics is a very important tool for genomic improvement. There is a need to continue collecting pedigree and phenotype information for standard traits and also to include novel economically important traits. It is needed to genotype animals for implementation of genomic selection. It is important to collaborate with other countries and different industry sectors. And we are doing studies at the University of Guelph and Omafra, and also in collaboration with other university and research institutes. And having the interest and motivation from the industry, it's very important for us because we want to work together and make some technology available that's going to increase your profitability. So this is the main goal of using genomics to increase the profitability of your flock. So thank you very much. And now I'm going to pass to Delma Kennedy, who is going to talk to you a little bit more. And at the end, if you have any questions, I'm very happy to answer any of them. Okay, thank you very much.
Thank you so much, Luis, for your great presentation. Now, in the meantime that we are switching presentations from French to Guelph, I'm going to introduce the next speaker. Is Delma Kennedy. Probably, well, I'm sure that all of you know her. She is a sheep specialist with the Ontario Ministry of Agriculture, Food and Rural Affairs. She coordinates the Ontario Sheep Genetic Evaluation Program from 1985 to 2010, and she continues to work with genetic evaluation projects as well as providing sheep extension information to Ontario sheep producers. Thank you so much, Delma. And yeah.
Put it on her. Okay. There we go. Okay, so I'm just going to talk a little bit about record keeping and genomics evaluations and what does this all mean in the future if we have genomics. So genomics, we just heard Louis tell us a bit about what it means, and there was a big to-do a number of years ago when the genome was mapped. So, I mean, 20 years ago when we were talking about DNA and mapping genomes, our big thing was once we get that mapped, can't we just pick the genes we want and clone the perfect sheep? Unfortunately, it hasn't really turned out that way. So this example here is just similar to what Lewis was showing you earlier. This is the callipege gene. And so this is a major gene was found on chromosome 18. And so the difference in a callipege animal, or one that has that muscling effect, is really one gene substitution. And so major genes were the first thing that was done. They're hard to find. We've also found out that they're relatively rare. So this means that it's a lot more complicated than we thought. We can't just pick the various genes and get the animal that we want. So most performance traits, a lot of what we're looking at, are controlled by many genes. And so when things are controlled by a lot of genes, it results in a continuous scale. When we look at the callipege, they either have it or they don't. It's not they sort of are callipege, right? And so if we use an example of feet or foot size, this first foot is small and the next foot is large. So if it was controlled by one gene, we tend to have a small foot or a large foot. But that's not what happens, right? What happens is we've got all of these feet in between. And so things are controlled by a bunch of genes, and they result on this scale where you see a gradual change from one end of the scale to the other. So many genes having a small effect produce performance. And that also means there can be a lot of different combinations that produce that performance. Another example is, why can you run faster than your brother? Even if you've had the same training, we're both out there, right, every day. Well, maybe your legs are longer proportionally to the rest of your body than your brother's. Or maybe you have a different ratio of slow and fast twitch muscles. Or maybe your ability to process oxygen is different. And so all of a sudden, same kind of thing, there's a whole bunch of different factors. And depending on which genes you've got for which traits, depends on the performance that we see. So the big problem then is we have to match genes to performance. And we have to know how every combination of those genes performs. And unfortunately, the only way we know what's going on is to keep records. So like Lewis said, we have to know what set of genes or what combination of genes is causing what performance. And you have to know it on a fairly large number of animals because there's so many different combinations that can happen. And so it takes a wide range of data to be able to sort out which combinations are giving you the performance that you want. Then we get another problem is that genetics are not static, right? So every time you breed an animal, different genes come together, you get a different combination. And so what happens is even if we do, say we get 5,000 animals, we DNA test them, we have the genome, we have the performance information, we know it's doing what, we're still not done, right? Because what happens is after a few years, we've got different combinations happening, a few mutations go on, and all of a sudden, the performance that we're seeing is not from the genetic combinations that we had 5 or 10 years ago. They're all slightly different. And so we have to keep updating. And so that updating thing, what does that mean? We have to keep keeping records. So we have to keep collecting the performance data. And so unfortunately, being able to identify genes doesn't ever get us away from that. So what we have here in Canada to produce genetic evaluations is Genovis. And so Genovis is a partnership of a number of groups. And so on the left, We have the Ontario Sheep Marketing Agency, Canadian Sheep Breeders Association, and the Centre d'Expertise en Production Ovine du Québec that all contribute funding to keep the genetic evaluation programs going on a regular basis. The Center for Genetic Improvement of Livestock houses the genetic evaluation program and the software to make it run. OMAFRA, we also put some resources now, just mainly in the form of my time rather than actual funding unless it comes through some specific projects. The database is housed here at the university to run the genetic evaluations. And the evaluations that you see, or EPDs, are all from the same system. So in Ontario, we have, there's a BioTrack program run by Beef Improvement Ontario, and they have genetic evaluations as well, and they come from Genovis. So their data is sent into that central database, EPDs are sent back to their system. So in Ontario, producers can access those evaluations by joining Genovis via CPOC in Quebec or by joining Bio with their BioTrack program. So if you join via CPOC, it costs about $250 a year, and if you want to take ultrasound measurements of loin depth or fat depth, there's extra charges for that. Just to give you an idea, the BioTrack program, I think, has a similar fee for their entry-level program, but they also have a sort of more extensive management aspect to their system, and they've also developed it so that you can use it on a phone or any device wherever you are. So one of the things that's happened here in Ontario with participation is over the years we've had fewer and fewer participants. So in 1993 we had about 275 producers, and currently we're running down about 38. I didn't put all of the Canada participation on here, but the rest, if I put the rest of Canada, well, this is actually all of Canada, including Ontario. So in 2008, there was 150 participants. Just over 50 of those were from Ontario, so about a third. And if you look at 2015, we're much less than a third now. So here, we're seeing much reduced participation. When we look at number of ewes in the early days, when we were getting fewer producers, we weren't as concerned because the number of ewes wasn't dropping significantly. It sort of stabilized through the early 2000s. But now as we're getting, more and fewer flocks, we're starting to see sort of a direct correlation between how many ewes we're getting recorded and how many flocks are left. So in all of Canada, it's not too bad. We're still testing around 20,000 ewes a year. But in Ontario, we're really losing a lot of participation. So that's very concerning for our industry here, considering that we have most of, the biggest percentage of ewes here in this province. So just another sort of angle with that is, everybody's used to talking about the pyramid where genetic change is top down, and we're doing our genetic improvement here at the top of the pyramid, and then the commercial industry is using those genetics to produce animals and produce marketable products. And so in 2015, when we're down to 38 producers, if we take the total number of animals that finished their 100-day test in 2015, and we assume that half of them are rams, so we divide that in half, that gives us 2,380 89 potential tested Ontario rams. That doesn't sound too bad until you think about the fact that we haven't culled them for anything, right? That's everything that made it to the 100-day test. We've done no culling on phenotype, on health, on anything. And so if we look at January 2015, we had about 20,000 ewes and replacement lambs that would need to be bred. And so if we assume that a ram breeds 50 ewes, then we would need 3,994 rams to breed these ewes and replacement lambs. And so if we also assume that a ram is in the flock for 3 years before it's gone, because then you're going to start running into inbreeding problems potentially, then we would need 1,332 replacement rams per year. So if we take that number, that 1,332, and divide it by the potential, then we'd have to keep the top 56% in order to satisfy that. And if we take that a step further, that includes all the breeds that are tested. So if we only look at the major breeds, which are Dorset, Suffolk, and Rideau, that are tested here in the province, if we just look at the number that finished their 100-day wait, that if we assume 50% are rams, there's only 1,213 of those. So if they were going to breed all of our ewes and replacement rams, we'd need more than we have. So when I used to do these numbers, even 10 years ago, we were looking at, we had to keep the top 20% of rams. And now we're into more than 50%. So it's becoming concerning about how we can make genetic improvement and genetic change in the province with the number of animals that are still being tested. And while we've been recording fewer animals, like Lewis talked about, this is an example from Australia, New Zealand, and basically all of these nice little digits here like BW for birth weight, weaning weight, 200-day weight, yearling weight, hogget weight, mature weight is what those stands for. These are all of the traits that they're currently recording information on and then doing genetic evaluations on. So you can see there's quite a few traits. When I started working with the sheep industry and genetics, which is unfortunately many years ago, almost 30 years ago, I guess, here in Ontario, we were sort of leading edge. We were running test stations, we were collecting information on traits and looking at information on individual animals. And so even though we progressed to doing genetic evaluations, in the last sort of decade to 15 years, we've rapidly started to fall behind in what we're measuring and the information that we're collecting on animals to be able to keep up with the changes in technology. And so here in Canada, we're currently collecting information on birth weight, 50-day weight, and 100-day weight, fat and muscle, and then on the reproductive side, there's number born, total weight, wean, survival, age at first lambing and interval. There's some research work going on in areas of health, parasites, and immunity. There's lots of opportunity, as Lewis said, in genomics and the resources here. But unfortunately, one of the things we have to have is data and performance data and people collecting records on these traits to be able to take advantage of these things. And so with genomics, yes, they will increase performance in hard to select for traits, but it is very expensive. Like, not only do we have to get those performance records taken and collected, we've also got to have DNA samples done, and there has to be a lot of DNA profiles of animals matched up with performance data. And so to make that work in our industry, we're going to have the industry doing more performance testing and collecting phenotypes before we can even start collecting much more on the DNA, so that we have something to match, because it's only effective if we can match things. In the past, with something like a major gene, it's fairly straightforward. You don't have to have you don't have to have the performance information to go with it, because it's one gene, one piece of information. You find it, you do the test, the animal has it or it doesn't. But the problem is most performance, or the bigger portion of performance, is controlled by a bunch of genes. And in something I was listening to lately, I heard someone saying that most of the time, for the performance traits that we're looking at, one gene will only explain less than 1% of what we're seeing. And so it's usually a large number of genes that are explaining the performance. And so sort of the bottom line is, the only way we're going to really be able to use genomics is if we can get more data recorded. And so in the future, some of the pressures we're seeing are environmental type things, health and welfare, efficiency. How do we get optimum output for inputs? And so we have to find, in order to address some of these things and keep up with what's going on in other countries and the rest of the world in the sheep industry, we're going to have to find a way to increase our performance testing and collect data for new traits that help us deal with these pressures. Otherwise, we're not going to be able to keep up. And the other big issue is that when we, in the past, you simply buy genetics, right? So, we import some semen and we make a change in our traits and sort of piggyback on the work that the other country's done. And the problem is, as we get into some of these lower heritability traits, it doesn't work that well, because some of the influences on the performance that we're seeing are coming from, the climate we happen to have, the environment we happen to have, the breeds we happen to have, what genetics are coming together and how they're coming together. And so we have to do some of this work ourselves in order to be able to leverage what's done in other countries and fit it together on how, and take advantage of some of the work they've done. And so that's all that I had. And so I guess we're to questions. Whether anybody wants to ask anything of Lewis or myself. I don't know. Do you know how to see the people? I don't know if we're muted. Can we unmute if somebody got? You might have to unmute your microphones. No?
The genetic testing for scrapies.
Yeah.
I know that's one example of the tests that are being done quite a bit in the U.S.
Yeah.
I don't seem to see it being done quite as much here. There's a number of other diseases as well that people are worried about with sheep. Is that something else that you think maybe we'll see more promotion of?
Well, the thing, I guess, with scrapie, it's basically, whether the animal has some resistance to scrapie or not. And so if you don't think you have any exposure to scrapie, then you're probably not going to pay for the testing. And so I think a lot of it with the scrapie is just whether people think it's worth investing the money to do the testing and having the resistant genotypes. if they don't think they're going to be faced with the disease, is sort of what it comes down to. And there are some producers who have done some extensive scrapie testing, but usually it's because they've had an animal that has come down with scrapie. And so they've, when they deal with CFIA and go into the program on how they're going to deal with the flock, is they usually end up testing the whole flock. And then depending on what genotypes they have, depends on what animals get put down and what animals are kept. And so that's sort of the thing with scrapie. And I don't know if you have any comments, Lewis, on something like, for example, the pneumonia work they did in New Zealand.
Yes, for example, for the pneumonia, what they were doing is they developed one score. So they would go to the slaughter place to collect the phenotype. So basically, they would look at the lungs and give one score. And based on those information, they would then create including the genetic evaluation and generate breeding values to select for that trait. So I think that there are several traits that can be used and can be added depending on, because each production system may be more interested in a specific group of traits or for a specific breed. So I guess like when define which traits you should collect phenotypes and which ones are important, it's relevant to define what's your breeding goal. So what you want to improve? Do you want to improve carcass weight, more reproduction traits, or you want to improve a combination of traits? So I think that you have to define what's your breeding goal, what are, let's say, the limitations, and what you want to improve. And based on that, that's the data that we are going to collect and select for.
So
does it answer your question?
Yeah, that's what I was going to say.
The reason I had asked is I just recently purchased 2 far superior rams to what I normally have, and I did tie up quite a bit of money in it. And hindsight being what it is, I wish I had them scrapie tested just so I would know for sure I was bringing in an animal that was adding to my traits in that more scrapie resistance. But at the time, I didn't really think about that, and I should have.
Yeah, yeah. And I mean, you still can test them, or I guess maybe you did test them. No, because you still, like, you can test them at any time for scrapie resistance, and then you know what you've got at least. Yeah.
Delma, just one comment that, for example, if you do a test, let's say for scrapie or like for any other trait, there are some tests, but most of those tests, the markers that they have, they are also including that technology that I mentioned, which is SNP shape. So it means when you are genotyping your animals using the SNP shape technology, those markers which are used to identify animals' resistance to certain diseases or to other traits, they are already included in this panel. So it means that when you genotype an animal, you are not only going to get the breeding values more accurate, but you are also going to be able to do parentage test if you want. You can estimate inbreeding of each animal. You can test and look for, like, in the case where there are already some major genes or some markers that are related to the trait, you can also identify that information when doing a genetic test. So there is a cost with this technology, but there is a lot of benefits as well.
And so what is the approximate cost? Do you know, Lewis?
Depends on, there are various SNP shape for sheep. There is, for example, there is one which is 6K, there is 12, there is 15, there is 50K, there is 600K. So the price depends on the amount of samples, animals that you genotype, and which SNP shape you are using. There are some of them around $40, There are some of them around $80, $90. So it all depends which there are more companies as well. So it's possible to negotiate, but I would say around like $50 to $80 per animal.
Okay. Okay. And so the big problem for a producer in Ontario or Canada right now is they could go and they could send some blood and get an analysis done, but then what would they get back or from who?
So, Delma,
But, but right now there isn't. Like, so right now they wouldn't actually be able to do anything with it until we had a project to pull some of that information together, because it'd have to be first matched with our performance data, wouldn't it?
yes, it would.
Yeah.
So
Yeah.
that's why I was mentioned that we do need to record pedigree, phenotypes, and genotypes. So those three
Yeah.
information are key. We need to have all three of them in order to be able to perform genomic selection. And there are a few traits which may have a test that may work, but it may not work for all breeds. So
Yes.
the key point, which I think to implement those tests efficiently, is to record performance data or for any trait that we are interested in. So it's important to have the phenotype in order to be able to use those tests.
That's good. Are there any other questions?
Delma, there is one here that was sent to the chat. That's from Bob Irvin. So the
Yeah.
question that he asked was, are you prepared to take DNA samples and using existing data from other countries for comparison? And the answer for that is depends. Because when we are comparing data, we need to make sure, because depends on which breeds we have data for. So, and what's the genetic connectedness between those two populations? So if we have, let's say, Dorset in Canada, and if we have data from Dorset in New Zealand, we may be able to work together because that's the same breed. There is a very high chance that they are going to be very genetically linked and that we are going to be able to work together. However, when we have populations which are very separate, very distant, in that case, we may not be able to combine data from different countries. This is actually the work that I'm doing here in France right now. So I want to see if it's possible to combine, in this case, that's for goats from Canada and France, to combine this data. And this methodology that we are developing here can also be applied for sheep as well. So the answer is yes, if the populations are genetically connected, genetically close.
Good.
It's Bob Irvin here. I'm just wondering, Luis, in our case, we have been using New Zealand semen for the past 15 years exclusively,
Okay.
and we also have been on Genova's forever. So, would you be prepared to accept some DNA samples and examine them and see just what markers are being exhibited here compared to what markers we requested from New Zealand when we imported the semen?
The animals that you import the semen, were they all genotyped, I assume, correct? And did you get this information? Bob, can you hear me?
Uh-oh. Did we lose him?
Delm, I think he's not hearing me.
Yeah, I'm just trying to see if I can find anything here, but we're kind of. Hold on, we're just seeing if we can see the people again. Can't we? Why can't we bring it up? We can't bring it up. What's that?
Delma?
Yes?
We're having connection issues at this end, I'm sure. I'm not sure if my oral question got through or not.
Yeah.
Okay, no, I did not hear your oral question.
Okay, so my question is, we've been using New Zealand genetics for approximately 15 years, and we've requested specific marker genes. I'm wondering if we sampled our sheep, could you evaluate what influence those marker genes have had here in this environment?
So what I would say is if you genotype your sheep, if you have the information, let's say if you record the traits and if you genotype enough number of animals, then yes. Or you can also, if you have access, because I'm not sure which group in New Zealand you were working with, they could also, let's say, if you send the samples to them, as they already have some of the data, and as you are importing semen from there, it means that your flock is connected to their flocks. So if you, they already have information, phenotypes and genotypes. So if you send your samples with the data that they have, you are going to be able to generate the molecular breeding values for your animals. But in order to do it, I know how to do it, but to do it, I would have to have access to the data.
Okay, so it is possible. The reason we have not been sending samples to New Zealand is it's cost prohibitive.
You mean like to send like DNA
Yes.
and blood?
What is required in terms of getting it into New Zealand is that we have to have the vet come. It's just a nightmare, a procedural nightmare to get the samples to New Zealand.
But you can also, for example, you can, the same technology that they genotype the animals in New Zealand, you can also genotype your animals in Canada and send all the data to them. And there are various laboratories in Canada that can do the genotyping. And there are some in Alberta, for example, and where you can do the genotyping and you send to them only the data, the information.
Yeah, Bob, on top of what Luis mentioned, here now across the street from university, we have a new facilities from GeneSeq that is very convenient. We can send here the samples. It's in Ontario. We don't need to send, to Alberta, to another place. And it's an international company that they do all the genotyping for livestock also in US and in Europe. So now we have one of the facilities here in Wales. So yeah, just to let you know that now it's even more easy, right? Because we have here, so that's another option we have now available. And all of the companies, they use the same technology and they are the same prices, so it's more or less the same. So.
Thank you.
Angela.
Yeah.
There is one question here from Colin where he asked, can dried blood card samples work? I don't know what he means by dried blood card samples.
Oh, yeah, I think. But for genotyping is his question?
He didn't mention, but I would say so.
Yeah, I'm not sure about the volume of the blood that is in these cars. So the only thing we can run, yes, they can send this and to extract DNA again, DNA is very, I mean, very strong, right? It's like we don't need special conditions to receive that. But the only concern I have is if is the enough volume of blood to can extract the DNA, enough DNA to run all the genotyping is the only thing, and I'm not sure now how much is the volume of the scars. But yeah, it's the only concern I have. Great. Any other questions?
Angela, I have a very quick question for you.
Yeah.
How do I get in touch with you? Because I have an email address for you, but it's not obviously getting through to you.
Yeah. Yeah. I'm always in touch. So, what we can do is, Delma, you have all your information, right? So Delma can send you all the email information for Luis and myself, and we are very happy to meet with you, to have emails, reply you, and any question that you have, we are very, very happy. So Delma can send all our contact information from Luis and myself, and yeah, so.
That would be great. Because
Perfect. I think that is the best way and more, yeah, perfect. Any other question? I think in 2 weeks we will do the next webinar.
Yes.
It's going to be also Tuesday, which day is?
At noon,
At noon,
March
the same time.
21st.
Yeah, March 21st. In the next webinar, we are going to talk a little bit about some of the research is going on here in the department. Focus on, again, some intestine, the parasite infection, and other research we are doing. Here in our group and in collaboration also with the OVC, the Ontario Veterinary College. And yeah, so, but again, we are gonna, Delma is gonna announce all the webinars, the next webinars and the topics we are gonna cover. And I think it was great. We have a great participation. 12 producers were connected today, and I think it was great to start, to move on and to be more in touch with you. Again, we need not to know what's the importance, what's the needs of the industry, what we can do to improve, and to show you all the technologies we have available now nowadays and all the research that we are doing to also to keep you posted and updated with everything. So I think it's great. So if we don't have any more questions, I think we can conclude. Yeah.
Yeah, sounds great.
Very good. So thank you so much and have a good afternoon, and see you hopefully in 2 weeks. Bye. Thank you, Luis, again for connected from France. Thank you so much.
Thank you.
Have a good night there. Bye.
Now, do we? Oh,
Yeah.
Bill is here too. Just
Coming to help you shut down properly.
Okay. Oh yeah, perfect. I press exit, but it was
You can just shut the
exit
computer
meeting.
down.
Exit meeting, yeah.
And for all.
Oh.
Okay, that's
Uh-oh.
fine.
Okay. So now it's okay. Should we? Okay.
Yeah, close it and shut the computer down.
So this I close here, and then I close here.
And then we can stop the recording here.
Perfect. And I think that's. Oh, I'm gonna.
shut
One
the
second. Delma, 1 second, 1 second.
Oh, I'm just gonna
Oh, okay.
pull out my.
Do you know about that part? You press that, and would you like to shut the room down? Yes.
Yes. Bye.
That turns the projector